Welcome back, biologists. This is Mr. Forberg, and today we're looking at another short free response question, this coming from the 2019 exam. I have a series of texts in front of me, so I'm going to straight dive in and let's take a look at it. So the text begins by saying that the yeast Saccharomyces cerevisiae is a single-celled organism. Amino acid synthesis in yeast cells occurs through metabolic pathways, and enzymes in the synthesis pathways are encoded for by different genes. The synthesis of a particular amino acid can be prevented by a mutation of a gene that encodes an enzyme required for the pathway. A researcher then conducts an experiment to determine the ability of yeast to grow on media that differ in amino acid content. Yeast can grow as both haploid or diploid organisms. The researcher tested two different haploid yeast strains, mutant 1 and 2, each of which has a single recessive mutation, and a haploid wild-type strain. The resulting data are shown in Table 1. So before I even get into parts A, B, and C, I want to take a look at Table 1 just to see what we're working with, and we can see that below here. So the first thing that I notice is here's my wild type, here's mutant 1 and mutant 2. All right, I know that the medium, we have 4 different treatments. In treatment number 1, all the amino acids are present, which means we should experience growth, and we do see that here as well as with the mutants as well. Treatment number 2, we have no amino acids present. That means that the yeast cells have to synthesize all those amino acids themselves. And so those wild type, all right, they're able to do that successfully, but these mutants have a mutation that's preventing them from creating one or more of those amino acids. In treatment number 3, we notice that all the amino acids are there, very similar to treatment 1, except we've removed methionine, so no methionine. And treatment number 4, they're all present, but now we no longer have a leucine. All right, so we've got a control, which is our wild type. We have 2 mutant strains, and we're putting them through a series of 4 treatments. The first is a positive control, make sure that everything's viable. The second is to go ahead and ensure that those mutants are unable to synthesize all the amino acids, and now we're looking for specific amino acids mutations in treatments 3 and 4. So let's get into the task verbs and the questions themselves. So part A, the first thing that we're being asked to do is to identify the role of treatment 1 in the experiment. All right, so I have to identify the role of treatment 1. And very simply put, as we scroll down, we said that treatment number 1 down here, we have all of our amino acids present. All right, and this is going to serve as a control. And there's kind of 3 things that you want to say. 1, this is certainly the control. It's trying to ensure that all cells are going to be viable. That means that both wild type, mutant 1, and mutant 2 are all viable cell lines. All right, and that's to make sure that any of the results from treatments 3 and 4 are really because of the treatment itself and not because of any other factors. All right, so we've answered part A. Let's move on to part B. In part B, our next task is to provide reasoning to explain how mutant 1 can grow on treatment 1, but not on treatment number 3. All right, so we're providing reasoning. Mutant 1 can grow on treatment 1, but not on treatment number 3. So let's take a look at treatment numbers 1 and 3 a little bit closely, or a little bit more closely. Now, part A established that treatment 1 is that control, so we know that mutant 1 can grow with when provided with all the right amino acids. However, in treatment number 3, we know that if we eliminate this methionine, we see that there's no growth present. So let's take a look at kind of what's going on in these metabolic pathways. Well, if we kind of refer back up to the top, and I'll annotate that in just a quick moment, we know that in order to produce a methionine, that particular mutant needs to make proteins. All right, there's an enzymatic reaction. So we're going to just call this a pre-methionine, and we've got to convert it to a fully functional methionine amino acid. In order to do that, we need the assistance of an enzyme. And so we're going to call this our methionine enzyme. All right, that enzyme is coded for by a gene. So in order to get this, we need a MET gene to be expressed to make our methionine enzyme, and that enzyme will help convert that pre-methionine to a fully functional methionine amino acid. So going back to our test question, or I should say the prompt itself, all right, we're trying to provide reasoning to explain why mutant 1 can grow on treatment number 1, but not on treatment number 3. All right, to do that, we know that there's a mutation in the MET gene, so no MET synthesis, meaning it can't produce that amino acid on its own because it can't produce the enzyme needed to catalyze the reaction. Now, because of that, if you can't produce all the amino acids you require, especially one like methionine, which is really important for the initiation of translation, you're not going to get fully functional proteins, and that cell or that cell line is not going to be able to survive. Now let's move on to the last part. Part C says yeast mate by fusing 2 haploid cells to make a diploid cell. In another experiment, the researcher made the mutant 1 and the mutant 2 haploid strains to produce diploid cells. Using the table provided, predict whether diploid cells will grow on each of the 4 media. Use a plus sign to indicate growth and a minus sign to indicate no growth. All right, so let's jump into what we really need to focus on. All right, we're predicting whether these diploid cells will grow on each media. All right, so let's scroll down. We probably figured out earlier that there was an extra column there. We weren't sure what to do with that. This is where part C comes in. So when we look at this particular setup, we know that we basically took this mutant and we fused them, and so now we have a diploid yeast cell that has both the genetic information from mutant 1 and mutant 2. Now, if we go back to treatment 1, this should be the easiest one. If we gave all the individual ones all the amino acids and they could grow, all right, having access to both genomes or both sets of genes should produce growth. All right, if we look, and I'm going to skip treatment 2 for a second and go down to 3, we knew that when we removed the gene or that mutation affected the gene from making the methionine enzyme, mutant 2 can still grow. It has that fully functional gene, but mutant 1 could not. But now that we're fusing the 2 together, all right, this mutant 2 functional gene is going to cover up the malfunctioning or the mutated mutant 1 version of the gene. So because of that, we would expect to see growth. In treatment number 4, we see the opposite. We see that mutant 2 has the particular mutation that's preventing it from making the amino acid leucine. However, mutant 1 has a fully functioning version, so it'll cover up that mistake. So when we fuse these 2 cells together, we still have a fully functional amino acid. And lastly, coming back to treatment number 2, all right, if we don't provide any amino acids, everything here has shown us that mutant 1 and mutant 2 have had all the right enzymes or the genes to create the enzymes to make the right amino acids, except for a single mutation in each. And because each of those cover up the other's mutation, all right, if we don't provide them with any amino acids, they should still be able to grow as well. So we would expect to see growth in all 4 particular treatment conditions. All right, and that answers part C for us. So pat yourself on the back if you've got all 3 of those. Thanks for stopping in. We'll see you next time.